Hello, my name is Nibal Khudeish, a doctoral researcher in the Neuroscience and Medicine Institute, INM-4. Today I'm going to give a presentation or an overview about my research project in neuroimaging epigenetics of resilience in post-traumatic stress disorder. So let's go to the introduction. Around 70 to 90% of individuals experience at least one traumatic event in their life, like child abuse, witnessing death or injury, sexual or physical assault, accident, or combat. However, there is a high degree of intra-individual variation in the way they react and respond to such events, where most of individuals can bounce back or adapt against such events and without developing any health consequences. And we can find that some of these individuals go to the opposite direction and develop a trauma related stress disorder like Ptsd or major depressive disorder. And one fundamental question is raised up here, which is why most of individual or some individual can overcome such a traumatic events while others are most susceptible or to trauma and stress, and go to develop such a trauma-related disorder. In order to understand this phenomena, we need to know more about our gene regulation and the processes that are related to or which are integrated in this. Our cells are composed of DNA, which is the genetic material of our body, and the DNA is as we all know, it is a chromosome. It's in the form of chromosome, and each chromosome consists of chromatin fiber. This chromatin fiber is composed of special subunits which called nucleosomes, and these nucleosomes consist the histone core and the DNA wrapped around this histone core proteins. In this construction, we can say that there's something called histone tails, which serve as a substrate for post-translational modification, or in other words, epigenetic modification, where some chemical tags like methyl groups or acetyl group, phosphorus group can be added, and by this addition, the cell can control the gene expression of some gene. This leads us to the epigenetic modifications. So there are three actually, but here I will mention only two of them. So the first one is histone modification. The histone modification, in this type of modification, chemical tags are added to the free tail of the histones, where we can find two kind of conformational changes, we can say. One of them is the heterochromatin, and which is characterized by the methylation on the histone, on specific sites of histone tails. In this type of a chromatin, the cell can switch off, or it switches off the gene expression. And while the second kind of chromatin is the achromatin, which is characterized by phosphorylation and acetylation. And in this case, the DNA, or the chromatin itself would be opened for gene expression, and the cell would, well, we can say that the cell is saying that go on for the gene expression in this situation. Another kind of epigenetic modification is the DNA methylation, where a group of or a methyl group is added to specific sites in the DNA called CPG island. So by this I summarized two of the epigenetic modifications in our body. And now if we go back to the scenario of post-traumatic stress disorder, one of the which represent one of the trauma related psychiatric disorder that can be observed in 20 to 30% of a individual exposed to extreme trauma. So one of the models that can show us what is happening in this disorder, that when an individual is exposed to specific or any type of stress or trauma, we can say extreme trauma, and this trauma will affect, or it's better to say it will trigger the epigenetic modification in specific cells. And this epigenetic modifications I just mentioned, like DNA methylation and histone modification, will affect the expression of genes which are implicated in stress responses, in neurotransmission, the neurotransmission in our brain, the immune regulation, and that's it. And the dysregulation of these genes mostly found in three systems, and due to the literature there are three systems which are implicated in the stress responses, which are the immunoendocrine systems, and based on the effect of a trauma on these three systems, many biological processes will be happens that time, and in the way that go either to resilience in the phenotype or to vulnerability in the phenotypes. So we can say that the all over epigenetic regulation of these three systems in response to trauma exposure appears to play a key role in indexing vulnerability versus development of PTSD. And in order to understand how an individual exhibit a resilient profile against such disorders, all these interactions need to be examined, and this leads us to our aims. So the aims of our study would be first to explore the gene-trauma epigenetic interaction that could affect the creation of a stress-resilient profile in a trauma-exposed individuals, and as well as this. As a second aim of our study is to study the structural and functional differences in brain connectivity between PTSD individual, trauma-exposed non-PTSD individual, and healthy controls. And the last but not least aim is to examine the associations between trauma severity, trait resilience, and PTSD. And in order to achieve our goals, we will follow the following methodology. So the first step in our methodology would be the participant. We will recruit 200, or we are aiming to recruit 200 refugees from the Middle East countries who live in Germany for sure. And these refugees should be between the age of 18 and 16 years old. And for sure, they should be interviewed, and they should follow, or they should meet and the inclusion criteria of our study in order to be included. So, in order to assess the mental health of these subjects, all the subjects will be diagnosed and interviewed by neuro or by psychiatrist from RWTH Aachen or Uniklinik, and using the DSM-5 criteria, and also they will be interviewed using the many interview on which the inclusion and exclusion criteria of the subject will be determined. The next step after the diagnosis would be the questionnaires. So in our study, we will ask the subjects who fulfilled the inclusion criteria to fill 3 sets of questionnaires. The first set is the resilience questionnaires, by which we can assess the resilience score for each individual, and in this set we will use another questionnaire for personality traits. So studies have shown that resilience is highly affected by the personality traits of the individual. And another questionnaire that we will use is the social support scale, in which we will assess how much the individual got social support during his trauma phase. And the last questionnaire in this list is a questionnaire for the well-being. And the second set of questionnaires includes some questionnaires related to trauma. The first one is the Harvard Trauma Questionnaire, which is mainly designed to study the trauma in refugees population. And the second one is the Life Stressor Checklist. It's a self-reported questionnaire where we can assess the type of stressful events that the subject went through in his life or her life. And the third one, which is related to diagnosis of PTSD. Actually, this is not a questionnaire, a self-reported questionnaire as the rest of a questionnaire. It's a kind of an interview by which we can assess the severity of PTSD, and also we can confirm the diagnosis of PTSD, the time of a trauma that the subject got exposed to. After this step, the subject will be screened in the 7 Tesla MRI scanner in order to study the structural and functional differences in brain connectivity between subjects groups. And we will focus mainly on some brain regions which showed an association, a neurobiological association of vulnerability to or resilience to stress-related disorders, due to the literature. And the last step in this methodology would be the epigenetic analysis. In this step, we will withdraw blood samples from all the individuals, and then we will extract the DNA. The DNA will be extracted, and the next step would be the epigenetic analysis using the biosequencing technique. And I should mention here that we will focus on studying or analyzing the DNA methylation of the individual, of specific genes and the individuals. So, and also one more important thing that I should mention here, that in this step will be done in cooperation with the Professor Cote and Professor Egerman from the genetics department in RWTH Aachen University. And after the data acquisition part, and the aim or the goal at this step would be to combine the epigenetic markers that we get from, or we got from the epigenetic analysis, and combine them with imaging data, because all the due to the literature, we've noticed that many epigenetic studies have been done before, and these epigenetics rely mainly on peripheral samples like blood or saliva, and it is insufficient to show what is happening in the brain. So in our study, or a new field is emerging now, which combined the imaging and the epigenetic together, and this new field called Neuroimaging Epigenetics, and we are going to use this field. And by using this field, we can combine the imaging data with epigenetic data in order to get a clearer image of what is happening or what is occurring in the central nervous system. And hopefully, using this approach, we will open new avenues to for Ptsd prevention or a treatment, and also for other stress-related disorders. And this is all what I have till now, and hopefully until the next presentation I can present more data, and I can clarify more about my research project. And until that time, be safe, and thank you very much.